next speaker is Dr. Andreas Fleischmann. He's the curator of vascular plants at the Munich Herbarium and research scientist at the GeoBioCenter at the University of Munich in Germany. His research on carnivorous plants includes evolution of carnivory in plants, systematics and phylogeny of carnivorous plants, especially in Drosera, Helianthera, and Lentibilariaceae. He has traveled around the world to see carnivores in the wild and has authored over a dozen papers on carnivorous plants, including the description of several new species. Also, he's grown carnivorous plants since he was a child. And he's written a few books, including the Genlysium monograph, which is an amazing piece of work. Andreas Fleischmann, our great honor to have him speaking.
Can you hear me? Yeah. Should we turn out the lights? That's for two reasons. First, you would see the photographs better, and second, I'm standing here and I wouldn't have to see Darren all the time here. Hi, Darren. So, let's see if this works. Perfect. So I'm going to talk about a topic which we will have several talks on, about the flower biology of carnivorous plants. Because so far, most of us have been interested in this part, and most scientists have been studying this part, so how carnivorous plants attract and capture prey, because this is fascinating and some kind of morbid. But I'm more interested, recently became more interested in the flower biology, because many carnivorous plants have surprisingly large and beautiful flowers, and especially, we know they attract some insects as prey, usually flying insects, but interestingly, all of the 800 species, more than 800 species of carnivorous plants we know, all of them are insect pollinated. None of them is wind pollinated. So they all need insects for food and for reproduction. That's why I chose the title, Food or Sex. So there's an apparent conflict for the plants. They have to attract insects to reproduce, but they also attract insects to feed on. And as we all know, some of the carnivorous plants, like the Sarracenia leucophylla or some of the Nepenthes species, this is Bocarensis, they look surprisingly similar to flowers, and they probably use attractants like scent and color, which are also attractants used by normal conventional plant flowers to attract insects. So insects could be attracted to the traps of this Venus flytrap or the flowers. So they have to choose. So is there something like a pollinator-prey conflict for the plant? Does it want to grow, get nutrients, but it also wants to reproduce, to produce seeds, to propagate? So how is this solved? And the Venus flytrap is a good example how early scientists have seen this. They usually explained they have large flowers and especially very long scapes. And this is still very popular, used in science and also in all the textbooks, that carnivorous plants have evolved long scapes to distract the pollinators from the deadly traps. This is one of the reasons why Brocchinia, for example, why Catopsis bertoniana was thought to be suspicious to be a carnivorous plant, because it has exceedingly long scapes which put away the flowers from the deadly traps. But if we look further in these carnivorous bromeliads, like here Brocchinia hectioides in the Gran Sabana of Venezuela, we have, of course, these tall scapes, very small, inconspicuous flowers. But there's some species like Brocchinia reducta, there's not much separation of the flowers from the rest of the plant. And for Helianthera, for example, I will show you some examples that these long scapes, they actually seem to serve more to make the flowers easy visible in the surrounding tall vegetation. This is true for many sundews, this is true for the Venus flytrap, and it's not for separating the flowers from the traps. This is not the case why they produce these long scapes. They use different techniques. In Helianthera, for example, famous on the tapuis of Venezuela, here this is Helianthera humantensis, which Thomas Carroll and others have even seen from the helicopter, because it produces such large clonal patches of red pitches, so this could be probably one single clone. And you see all these flower spikes sticking out. They have very tall, long flower scapes, but it's not for separating the pollinator from the traps. It's for making these flowers more easy visible. They have huge flowers with 4 tepals, which look like petals. They're usually hanging down, like in many plants which grow in high rainfall climates to protect the sexual organs, so the anthers and the gynecium, from the rain, hanging down. And they are, so that's how to do field work on the tepuis, arriving with a helicopter, having just 15 minutes time for field work before departing again. If you don't have a lot of money to spend, rather time on the tepuis paying the helicopter. And as we probably all know, or those growing Helianthera, you're welcome. If you cut open one of the flowers, in Helianthera, there's one particular thing which is typical to Sarraceniaceae, that the flower organs do not ripe at the same time. This is a flower at the male phase. The flower starts in the female phase, when here the style, the pistil is ripe. This is already dry and brown. And now the anthers are ripe. This is for the plant, there's nothing special about carnivorous plants. This is just to avoid self-pollination. It's dichogamy, so to separate the sexual organs. They're first females and then so-called transgender, they are getting male. And the anthers are also special because you probably know if you try to pollinate them, it's not like in Sarracenia that they split open and release all the pollen. They have little tiny pores at the apex, and they do not fully split open. They have to be shaken in a special manner by large bees or bumble bees. They are bus pollinated like tomato plants. And these are here large carpenter bees. Sorry, here. Large carpenter bees of the genus Xylocopa. They're also native here to the US. And they buzz vibrate these flowers. Flowers of Helianthera do not offer any nectar. And the bees, they can well discriminate. I just go collecting for pollen. So they know these white flowers, I just go there for pollen. They are at this time not interested in nectar. So here you see these bus-pollinated flowers. Sometimes you even see scratches where they did hold on with their claws. But they're only interested in pollen, not in nectar. So these kind of bees are not attracted to the pitcher leaves at all, because the reward which the pitcher gives to the insects, or the deadly attractant, is nectar. They are only interested in pollen. And the only case of pollinator-prey conflict where I ever have seen where a carnivorous plant has been eating its potential pollinators is this one. And this is not a native species. Here, this bee, this is Africanized honeybee, or killer bees like they're called. They are everywhere in South America, even on the tepuis. And they frequently feed on the nectar of the traps, and also try to collect the pollen. Honeybees cannot bus pollinate, so what they feed on is this pollen spill, which is left over by the large carpenter bees, and they collect this pollen to bring it to their nest. But they also have to collect, like all other bees, so pollen is the food source, the protein for the larvae, and nectar usually in bees is their own nutrition, so that's the fuel which they consume for energy. And here it's trying to collect some pollen, and afterwards it's trying to feed on. This is here on Helianthera sarracenioides, for example. And you see it's carrying Helianthera pollen. And now it's feeding on these large nectaries. And here you can see sticking Helianthera pollen everywhere on this trap. And they are regularly found. I've opened some of these sarracenioides pictures, and they were full with Africanized honeybees. So this is a case where there's not a natural pollinator, and so there's no adaptation to discriminate between flowers and this food source. So for the plant, these honeybees have no benefit at all, because they're first carrying some pollen, but then are fed, so all the male organs are wasted for the plant, and it's just a little bit of nutrition. How does Sarracenia do? If we have these huge stands of Sarracenia, I'm just briefly focused on Sarracenia because I know that Larry will have a beautiful talk tomorrow on this, and I have not been during flowering season in Sarracenia habitat, so my photographs are from cultivation. But one thing which you probably all know when you grow Sarracenia, the time they flower, they usually have not present many freshly open pitchers. They usually, the pitchers are still closed or just at the state of developing. The flower of Sarracenia is also very peculiar. If we cut it in open, it looks very strange. It's a bit confusing. These flower organs, they have bracts. The actual sepal, which looks like a sepal, is actually a bract. These large things, they are the sepals, the petals hanging down, quite short-lived. And then this umbrella or stylar disc. These are 5 fused styles. Here's the pistil, these 5 styles. fuse to form an umbrella. And this serves, and here are the anthers hanging down. In contrast to Helianthera, they split open, and the pollen is shed on this stylar disc. This is what botanists call secondary pollen presentation. So the pollen is usually not collected from the anthers, but from this stylar disc. It's still well separated from the stigma. Here, the stigmas are outside the flower. This is like a closed basket, which are closed by the petals. The stigmas are outside, the pollen is inside, not much can happen. And pollinators have to enter, they usually land on the petals, climb in there, and to reach the pollen and the nectar. In contrast to Helianthera, Sarracenia produces nectar, and while doing so, they pass this stigma. So it's just this little thing. I think it's not necessary to tell you that you do not need to put pollen here, but pollen here to fertilize, to hybridize Sarracenia. Well, here's a flower in my greenhouse, and bumblebees are very smart. You probably know this. Bumblebees learn all different flowers. Even untrained workers, they just experience how this works. So even our native European bumblebees, here's a garden bumblebee, they know how these flowers work. They land here on the petal, they climb in. You see this is pollen. It has collected from different flowers, but here it's dusted in Sarracenia pollen. They crawl in here, touching the stigma, and then inside I opened this flower and had to wait for a long time until the bumblebee was interested in this artificially opened flower. And while feeding on the nectar and collecting pollen, it's either collecting pollen from the anthers, then it gets dusted with pollen on its back from the pollen on this stylar disc here, or it's collecting pollen from the stylar disc and it gets pollen on the back from the anthers. So the plant has no interest in the bee collecting pollen because the plant offers nectar as a floral reward, but pollen is actually for its own reproduction. So it tries to avoid that the bee discovers, or the bumblebee discovers, that it's dusted in pollen. And when it leaves out, it cannot, this way is closed, so it has to lift the petal. There's a little bit of light shining through, and it leaves without touching the stigma anymore. And so this is mechanic isolation of the two organs. This, of course, does not prevent pollinator-prey conflict, but nevertheless, I think it's interesting. About pollinator-prey conflict, Larry will talk tomorrow. How about Drosophyllum, a Mediterranean shrub from Spain, Portugal, and Northern Africa? The dewy pine growing in comparatively dry environment, very sticky, and again, you can see the flowers are not well separated from the leaves. They are found amongst the sticky carnivorous leaves. And here again, it's different attractants to prey and pollinators. These plants in natural habitat, you see how they are glistening, how they are shining in full sun, and the leaves mimic nectar. So the insects which are attracted to the leaves, they are foraging for nectar. They want to feed on nectar droplets. It's pollinated by large bees or beetles like this scarabate beetle, and the flowers, again, they have no nectar. So there's no confusion for the pollinators. They exclusively go for these yellow flowers to go for nectar. This is a common pattern in this Mediterranean area. There are also some species of cistus, rock roses, I think they're called in English, which also have yellow flowers and no nectar. And so the pollinators learn this syndrome, the Floral Guild, how it's called. I have no photos of Beetles pollinators because it's very rare that they are observed. Beetles, again, has its flowers amidst the sticky carnivorous leaves, no matter if it's Beetles lamellata or gigantea or Beetles liniflora, the flowers are found amongst the sticky carnivorous leaves. So please, for those who write textbooks or school books, forget about this. Carnivorous plants have long scapes to avoid having their pollinators being trapped. It's nonsense. They have different techniques. Again, in Beetles, we have anthers which are closed. They do not open. Here is the style, and here are the anthers. And they, again, have to be shaken in the correct way. They're bus-pollinated. And again, bus-pollination can only be done by large bees, bumblebee size, or a bit smaller. So any insect which is visiting these flowers is not very likely to stick to this very weak glue. The captured insects are usually much smaller. They do not serve as pollinators. So they really distinguish well between pollinators and food. Most famous example probably of pollinator-prey conflict is Roridula. And in this plant, again, here we have Roridula gorgonias from the wetter parts of the Western Cape, but also in the drier part. here Aurigula dentata, you see that the flowers, they are not on very long scapes. They're very big. And again, in Aurigula, the flowers have to be buzz pollinated, at least in Aurigula dentata. And my colleague Bruce Anderson who has been studying this plant for decades now in in South Africa, found almost no pollinators visiting these flowers. It's expected to have large bees, but interestingly, the mucilage, this resin of Aurigula, it's very strong, it's very sticky. You probably, if you grow this plant in your garden or your greenhouse, there are yellow jackets, wasps, hornets, even large bumblebees. There are occasional reports of small birds sticking to this plant. So even the large pollinators, which would normally buzz pollinate, they could be potentially trapped by this plant. So this is really a dilemma. So how does this plant, this seems to be an evolutionary dead end, but somehow it must work. And Bruce Anderson found out who's the pollinator. As you probably all know, Aurigula has these mutualistic bugs of the genus Palmaridea, which live on the leaves, feed on the trapped insects. And they are also frequently found in the flowers, especially the juveniles. And they are piercing the flowers, feeding a bit on the pollen, usually get dusted on pollen and touch the stigma. And this is really an evolutionary, somehow dead end, because the only thing these bugs are now doing, Aurigula cannot self. It has to be pollinated. This mechanism with the buzz pollination avoids self-pollination. And what Bruce Anderson calls this is a mutualist mediated selfing. So these plants are apparently always selfing in the wild, because these bugs do not fly for long distance, so they do not carry pollen, especially because it's usually the larvae or the nymphs which do not have wings developed. So apparently even in the wild, Aurigula is usually predominantly selfing. Genetic studies show this, that the population usually is very uniform genetically. So this could be the only case where there's an apparent pollinator-prey conflict which is solved, but this plant feeds on everything which is approaching, even the flowers, and that these little critters which live on the plants, they help to self the plants, and from time to time probably they manage to crawl from, to move from population to population to enable some genetic exchange. But this is really a very curious and strange case. So for Pinguicula, it's very obvious. These huge flowers attract a lot of insects, and again, we can see already from size relation, these are flowers which are probably attracting large pollinators, while usually smaller things are trapped on these sticky butterwort leaves. Pollinators of these flowers are usually bees or large flies or bee flies. I was fortunate enough to see natural pollinators of Pinguicula vallisnerifolia in Spain. You see this is a small mining bee of the genus Andrena, which carries pollen, this whitish pollen of Pinguicula on its back, leaving the flowers. This is Pinguicula pollen, and which will deposit the pollen on the next flower. There are also some species where it's, I think, very obvious which kind of animal is pollinating these flowers. They are hummingbird pollinated. Some of the Mexican species like Pinguicula laoiana, where you can see, first of all, the red color. It's always a good indication that this is hummingbird pollination. And second, you can see even how deep the beak is inserted. This is the part where the spur is widened, and then the spur is suddenly narrowed, and only at the very tip you find a lot of nectar. And the beak will be inserted just right to this place, and the rest is then occupied by the tongue of the hummingbird, who sucks out the nectar. And if you remove nectar from this spur or a non-hummingbird pollinated flower, you will notice that Pinguicula laoiana or similar hummingbird pollinated species will refill the nectar much more frequently, and they contain much more nectar because hummingbirds consume much more nectar. It's also a typical syndrome found in hummingbird pollinated flowers. Compared to this one, this is certainly, Pinguicula colimensis is certainly not pollinated by hummingbirds because the spur is curved, and hummingbirds cannot insert their beak here. This also contains only a little bit of nectar droplets, which is not continuously refilled. And this, we know almost nothing about Mexican Pinguicula pollinators because Fernando always just goes to the wall, makes a selfie of him and the plants, grabs a few herbarium specimens, but he's usually bored at one side quite quickly. So for pollinator observations, you have to wait there for hours. And so, how many pollinators photos do you have? You see. How many species have you seen in the wild of Pinguicula?
I don't know.
Probably all 100. So it's about the relation. So it's probably butterflies. This is a landing platform for butterflies or hoverflies to sit here and insert their proboscis. And I assume I would predict it's butterflies because the spur is curved and only butterflies can curve their proboscis, while in bee flies it's straight. Very interesting pattern we find in the southeastern US in some pinguicular species is this strange organ protruding here, and I know some people have written to me, I'm pollinating pinguicula primuliflora, and I'm dabbing here, get no pollen, where's the pollen? It does not work, they do not set seed, because this is not the pollen, this is not the stamens, this is false pollen, this is an outgrowth from the corolla, which is mimicking pollen. We find this in a few Mexican species like Pinguicula departiana, which has this sterile outgrowth here, which looks like pollen. But if we make a cross-section of the flower, you can see this is the false pollen. The real pollen is here. They have two anthers, and they have a stigma which covers the anthers. It's again mechanical separation in Pinguicula, like in most Lentibulariaceae flower. Here we have the spur with nectar. So here we have the real pollen, and here we have the false pollen. And we find this in many Pinguicula species to distract pollinators from the real pollen, not to collect and feed on the pollen. So it's trying to collect this pollen and gets dusted with the real pollen on its back, which it does not notice. The plant is hiding its pollen there, and the plant gets to the next plant as well. This is very common, this syndrome, in the southeastern United States, where also some orchids, like I think Calopogon, I don't know the English name, something with pink, meadow pink, probably. Common names, yes. So Calopogon, it uses exactly the same, and for Calopogon it's known that it's usually untrained bumblebee workers. When they're freshly hatched, they don't know what real pollen looks like. They will go for everything which is bristly, yellowy, and looks a little bit like pollen. So, and they quickly learn it and try to avoid these wrong flowers. So here you see, for example, a daggerfly is trying to feed on this wrong false pollen on Pinguicula leptoceras in the Austrian Alps, and then suddenly realizes this is not the real thing. It follows these nectar guides, gets some nectar, and then gets the pollen dusted on its back. And again, in Pinguicula, in all cases we know the pollinators are way too large to be trapped on the flowers. Actually, I always make these experiments, and if I see a pollinator, I trap the pollinator, stick it on the leaf, and it can usually escape. Yeah. Beetle, you make herbarium specimens by thousands, so... Okay, in Reticularia, it's very obvious that we have no pollinator-prey conflict because we have separation by trapping and reproduction. The one part is underneath the soil and the upper part is above soil, so it's well separated, of course, and the prey is way too small to be pollinated. But again, we know surprisingly few about Reticularia. There are about 250 species of Reticularia having been described by now, known to science, and we know almost nothing. There are 2 publications about pollination biology of Utricularia. We have huge flowers, like here in Utricularia humboldtiana, which is growing here, scenic view of Mount Roraima. We have mass flowering, like in what was formerly known as Polypomphylix, Utricularia multifida. And the problem is, why there are, I think, so few observations on pollination of Lentibulariaceae, you have to wait a long time here, and usually you get wet feet. So that's probably one reason. But with Thomas Carroll in Western Australia in 2008, we were lucky enough here, we took our multifida. Thomas took these beautiful photographs of this small sweat bee, so it's family Halictides, and again it's visiting the flower and get pollen dusted on its back. And again, interestingly, we have these false pollen, these rims which are yellow and very conspicuous. This is something which we usually find in Utricularia in Australia. So there has to be something here in Australia where the pollinators probably usually tend to collect pollen from the flowers. Again, we could guess here on the tapuis in Venezuela which kind of animal is pollinating Utricularia kelchii. Again, I think it's hummingbird pollination. I would pay a large box of beer for anyone who's showing me a photograph. I predict this is hummingbird pollinated. It perfectly fits. The spur is straight, the nectar is curved here, so the tongue bends down here. The beak is feeding in here. Same for Utricularia campbelliana. You see these flowers, they do not have a palate which is closing here. The flowers are open in contrast to many other Utricularia species because the beak could not work here if the flower would be masked or closed. So it's very likely hummingbird pollinated. This is one of the species here. Again, we moved to Western Australia, where pollinators are known. This is Utricularium incesii, and if you ever have wondered why it's red and has such an incredibly huge spur filled with nectar, this species is pollinated by nectar eaters, so birds, and it's even indication that they mimic flowers of Grevillea species which are around and also red. Robert might be the one to talk to. Have you ever seen birds on these flowers, Robert?
No, I haven't.
Okay, yeah. Because there are some, yeah, and some Utricularia species fascinating. What you see here in this piece of mud is barely nothing. This is a close-up of Utricularia simonsii, just recently discovered, described in 2008 by Al Nauri. And this Utricularia has this Utricularia has the distinction of having the smallest flowers with a fully developed flower. The smallest flowers still are duckweeds, but then the flower is reduced to a single stamen or a single pistil. And here we have a fully developed flower. We have a corolla, which is bilabiate. We have here a stigma, two anthers. Everything is functional, and it's just tiny. It's maybe half of a millimeter, or maximum 1 millimeter. And interestingly, there the flowers are open again. They do not have, in most Utricularia species we have masked flowers, so that we have this upward washing palette, so this gibbous thing which is pressed against the upper lip to close the flowers to hide the sexual organs. The smaller we get in Utricularia, we have open flowers, like in Pinguicula. And for me, the explanation is because it would mechanically not work. The pollinator here has to be so small that the forces to open the flower, if it's a masked flower, would be too strong. So all small Utricularia species, interestingly, have open flowers. But still, this thing, I have it in cultivation, has to be hand-pollinated. If you don't pollinate it, it will not send seed. So it's not a weed like Subulata, which is self-pollinating. You have to pollinate it to get seed. Same for this one, Utricularia. There's a whole complex of species, I think now 7 species of this Antinifera or Capilliflora complex, with strange outgrowths. In some species it's the upper lip which has these long appendages, the lower lip is fringed, so it looks almost like an alien, looks like an insect, and we don't know anything. Peter Taylor just described the flowers with these appendages. Peter Taylor has a very delicate language of describing flowers. For example, he called the shape of this here, of the spur, it's scrotiform. And all of these species have these scrotum-like spur filled with nectar. The corolla is open, and we don't know, is there any insect waiting? I was waiting for a single day here in the field, almost in front of these flowers. The only thing that came were mosquitoes, but they were attracted to me, not to the flowers. And some sweat bees, which also were attracted to me, not to the flowers. A student of mine, Thilo Krüger, spent 1 day and 1 night in the field, nothing. No pollinators. They have a sweet scent, which is, because there was indication, Alan Lowry was proposing that this could be a sexual mimic, like some bee orchids, some of the genus Orpheus. But we don't know the pollinators. So again, this would be chance for field work. Beautiful area in northern Australia. Mosquito time was, yeah. The only thing is you have to find these. This is one of these amazing flowers, and here's my finger for scale. I'm from Munich area, so southern Germany, and we usually have quite fat fingers, but this is real scale, so I don't have fat fingers. These are tiny, and they are hidden amongst vegetation, so they're really difficult to spot between all these dry grasses. So whatever is the pollinator, it really has to be attracted really effectively to these flowers, because again, they have to be in cultivation. I know this from cultivation, they have to be hand pollinated, they're not selfing. So when there's a huge seed set in the wild. So again, time for field work. If you have students, if you have time, if you are out there, just again, I would pay a lot of money. Usually in Germany we pay beer as a price, so I would pay beer or whatever you prefer. Genlissia, one of my favorite genera. Again, the flower very similar to Utricularia. Actually nothing special about this, but fascinating, we have 2 subgenera in Genlissia, subgenus Tayloria and Genlissia, which are separated by how the seed capsules open, by phylogeny, but also, which nobody noticed before, by the flowers. Because in subgenus Tayloria, Genlissia subgenus Tayloria, the spur is spreading from the lower lip, so it's parallel to the pedicel, while in subgenus Delicia, it's parallel to the lower lip, so it's bent around. This thing looks more like a peduncular flower, this thing more like a utricularia flower. And here we have so-called salvariform flowers, or snap, and this is a snapdragon type flower with this enclosed thing. So here we have an open tubular corolla throat, where whoever wants to enter to reach the nectar at the tip of the spur can freely enter and freely access the stamens, whereas here this palate is firmly enclosing it. Here we have the reproductive organs, here we have the nectar, so everything is hidden inside. So whatever is entering this is probably sitting here on this palate and inserting its head or proboscis, wherever, and here we have to have stronger insects which have to push open the flowers or pull open the flowers. We don't know anything, or actually we now know something about this because there is a student of Vitor Miranda in Brazil who studied the pollinators, and it's actually indeed bee flies, flies and bees landing here on this platform, on this flat platform, inserting their head and proboscis. While I was fortunate enough in Sierra Leone to take photographs of this pollinator. Don't worry, I was there in 2006, so way before the Ebola outbreak. So I'm not hazardous. And it's again here small sweat bees pushing open the flower to reach with the tongue. You probably can see it here with the tongue, the nectar. So I hope I still have time because we come now to the most fascinating genus, not only in terms of diversity in biology, but also in terms of flower biology. But I also will keep this briefly because Robert Gibson will talk about this later tomorrow. Just a few facts. We have about 250 species. They occur worldwide, and the redder the color gets, the more species we have. So most people who study sundews live in the northern hemisphere, but the diversity is here. That's why I love studying Drosera, because this is usually our winter here. You can go there and study the plants in the field. But interestingly, most scientists who work on carnivorous plants are here in the northern hemisphere, and that's why I marked where studies on Drosera flower biology had been taken out. So they had been carried out in Europe, of course, mainly Germany and England, some in the United States and in Canada, and one exception here by a German in New Zealand. What did they study? Well, in Europe they studied the 3 species of Drosera we have there, the long-leaved, the intermediate, and the round-leaved sundew. In terms of flower diversity, quite boring. In the United States they added one which is spectacularly different, Drosera linearis. And in New Zealand, they chose New Zealand because they have here 2 species, Drosera spatulata and Drosera arcturiae. All have small, selfing, white flowers. And this study particularly is interesting because they took all the literature reviews and said, Drosera has small flowers which are selfing. Not much pollinator prey conflict, they're selfing small white flowers. They have here, 6, 7 species and generalized for the whole genus of 250 species. That's the difference between an ecologist or a systematic botanist, who I am. So they should look here in Australia. Australia has more than 150 species, 107 according to my latest counts, in Western Australia alone, and look at this huge diversity of different flower types. Colors, different flower forms. This is an area you should study pollinator and pollination biology of Drosera. Another thing, in Drosera again, it was long argued that they all have long scapes. In most textbooks you find all Drosera species have long scapes to separate pollinators from prey. So that this is food, this is sex, and that we have a spatial separation of reproductive organs. But I will show you some contrary cases. Some of the tuberous Droseras, like here Drosera rupicola, which comes in different color forms. The flowers are almost nested amongst the sticky leaves. Or here in Drosera ramelosa, also the flowers not very much separated. In many of the climbing tuberous Drosera, the uppermost sticky leaves, the flowers sit almost amongst these leaves. And to the more extreme examples, one species which Fernando discovered, Drosera amazonica, the flowers are sessile, they have no stalk at all. Drosera solaris, Drosera arenicola, Drosera felix, and photos from Fernando, Drosera meristocaulis. So there is no spatial separation, and still they do well, they produce seed. It has nothing to do with affinity, because in this section, which has two members, this is Drosera uniflora and Drosera stinopedala, one has long scapes and one has very short scapes. This grows in open windswept areas on the Falkland Islands, for example, or the Magellan Strait, in sphagnum carpets. This grows also in similar habitat, but usually in more dense vegetation. So for me, a very good indication this is something to lift the flowers above the surrounding vegetation, but not to distract pollinators. Again, here Drosera arcturiae, similar habitats, and here the flowers are usually found amongst the leaves, like in our Drosera intermedia. There are some species which have very tall scapes, and I'm currently working on a study. Why do pygmy Drosera and other Drosera have long scapes? In pygmy Drosera, I think it has to do, when they flower, where the soil gets very hot, and flowers of Drosera are very heat sensitive. I discovered this in the greenhouse. When the flowers come very close to the light bulbs, they will burn or just develop crippled flowers. And the flowers on the ground, it's very hot. Many pygmy flowers flower already in the dry season when they have almost no leaves developed anymore. They flower from a stipule bud, and they lift the flowers above the hot air from the ground. It's just a few centimeters, but it's enough. And if you live in a dry climate, you will see during the day where all the snails go. They climb up little pieces of grass they do not rest on the ground because it's getting too hot, and I think they also expose their flowers above ground. And this is something Australian students in a corporation will try to discover. You can easily find it out with fieldwork. Just bend down the flower scapes on the ground, let the flowers develop on the ground, and see what happens. Some Drosera species, again, very short flower scapes, but some of the tuberous species, they have again a temporal separation of flowers and leaves. So here it's like almost in crocus, that the flowers develop long before the sticky carnivorous leaves develop. Here, for example, in Drosera laureae or Drosera rosulata, here you see a sticky leaf, a rosette of sticky leaves, but this is a spent flower already, while this one is putting up a new flower, and you see the leaves are not fully enfolded, not very functional, so there's also no pollinator-prey conflict. In others, like Brazilian species, like here Drosera chrysolepis, again the leaves mimic, like a Drosophyllum, mimic nectar, while the flowers have no nectar. Almost all Drosera species are nectarless. They offer no nectar, only pollen. So the pollinator goes for the pollen, and whoever is attracted to this as prey goes for nectar. So what about the pollinators? Pollinators are very diverse in Drosera, but it's three main groups. One large group are flies, especially flower flies or hoverflies. Here in Drosera gigantea in Australia, Drosera lasianta, you see a flower fly dabbing on the pollen here. Flower flies are among the very few animals which can digest pollen, fully digested. But also other flies, here in Drosera verrucata, there's a species of blowfly. Here another species of fly on Drosera nivea. And this is one case where I need to ask the Australian experts. They are very effective pollinators. I've seen them carrying pollen from flower to flower, but I do not know if it's the native flower pollinator, because I don't know which fly species were introduced to Australia and which are native, but I think Robert can confirm that flies now are the most abundant animals in Australia, right? So maybe they just found something which they could adapt to, and they're not the natural pollinators. This flower has no scent, but several pygmy Drosera have a very strong scent, yeast-like heavy scent, like Drosera verrucata, which is very attractive to flies. Again, flowers here in the members of the Drosera indica complex, very closely presented close to the leaves. This is Drosera finlaysoniana, one of the species which sometimes has sentient leaves, like the dewy pine. And here, Alice Robinson took photographs of bees. Here it's again a halictic bee, which is collecting pollen from the flowers. So apparently here again the floral reward is pollen. While the insects which are attracted to the flowers in huge numbers, they go for nectar. These are butterflies, for example. Butterflies do not go for pollen. They cannot feed on pollen. They have a very narrow proboscis. They cannot suck up pollen. It would like to be trying to eat a dumpling through a piece of straw. That does not work. So they just go for nectar, and they are attracted in huge numbers to some of these sentient Drosera species. both day-active butterflies, nocturnal moths, and also small insects like thrips, for example, make up a high percentage of these insects. And I've studied dozens of herbarium specimens and specimens in the wild of the members of Drosera indica complex, screened them for prey. There was not a single bee found on these flowers. So again, probably very well separated. So we have flies, bees, and the next largest group of pollinators of Drosera, beetles. Very common, especially in Drosera species which have flower contrasts. So bicolored flowers, this is always a good indication of beetle pollination, because beetles, they go for strong flower contrasts. They do not go so much for scent or anything which is glistening, but for flower contrasts. You probably know this. If you would go out in summer with a yellow jacket, or the yellow sweater, you would attract tons of beetles, because they're attracted. Blue sky and yellow shirt, this attracts beetles. So they especially go for flowers which show high contrast. And they are also frequently found both in Australia. Here we can see a photo of Drosera thysonosepala, or Menziesii, member of the Menziesii gang, where this monkey beetle or scarab beetle is carrying pollen. In South Africa, the same. They feed on pollen. They are very ancestral pollinators, beetles, so they usually ruin the flowers. They feed on the flower parts, on everything, but they are very bristly and carry some pollen. So in Australia and in South Africa, they're called monkey beetles. We also sometimes find here Setocorus, so these mutualistic sundew bugs, piercing flowers, feeding on the pollen, also carrying some pollen, but they're not very active pollinators because they usually don't leave their host sundew, so they only walk around on these flowers, and in most tuberous sundews, they're not helping with pollination. So usually in the flowers, no nectar. On the leaves, mimics of nectar, so works fine because Drosera flowers have no nectar, with a few exceptions. They have no nectaries, but in some sundew species, exactly only in a few tuberous sundew species, all rosetted tuberous species, we have nectar droplets on the styles. These are stigmatic exudates, where Alan Lowry assumes they aid pollination, like in gymnosperms, as a pollination droplet, but that is not the case. They are high sugary content. I measured the glucose content, and they're very sweet, high sugar content, and they're consumed by the pollinators. And Alan Lowry already depicted this without knowing in his books, in his textbooks, for the members of the Solonifera complex. He said some of them have dimorphic styles, and he drew it in like this here. There's like a brush of styles and the crown surrounding them. And if you look at Drosera stolonifera, these dimorphic styles have two different functions. These are the normally developed styles, which can take up the pollen, and these are sterile styles. I tested this in cultivation. You need two different clones in the stolonifera complex. If you put pollen here, no single seed grain will ripen. So these styles are modified to become nectaries, and they produce nectar droplets here to offer to the pollinator. And again, here, a pollinator which is going for nectar is this moth, for example, and it's feeding on the nectar from the styles here in Drosera fimbriata, which has the same technique. And you can probably see here in the first rows, there's pollen sticking to its proboscis. So another thing is color patterns in Drosera flowers, which fascinates me. You have to go to Australia to work on Drosera flower biology. You have a variety of flower colors. Some look very similar, and some of these species look very dissimilar. And now the thing is, are these related, or by phylogeny, are these related, these species? So from morphology, it indeed indicates that not all the orange things and all the pink things and all the red, white things are related, but all with the same style shape, for example, which I drew here in, is a style shape, and there's also indication from phylogeny that it's not this species which is related to this one, but these are related. But regarding pollinators, it's of course that these share probably the same pollinator, and these share the same pollinator, and so on. Which is also interesting. What we find in some Drosera species are so-called pollinator guilds or floral guilds, that all these flowers look similar, but not only among Drosera, but also among the nearby growing Stylidium species. So what is growing here together all has white flowers with a red dot. And there's sometimes different structures which make up the red dot. Here, for example, it's the ovary and the base of the tepals. Here, for example, it's the styles which make up this red dot, but the pattern all looks very similar. So I would assume that it's very likely that they all share the same pollinator, but probably do not hybridize because these things are not very closely related. Also very interesting, if you look at some of these members with bicolored flowers, as I said, some plants try to avoid that their pollen is being collected, especially in pygmy Drosera. They produce very few pollen, so they produce something to distract the pollinator from the pollen. And you see probably these shining areas here. This is Drosera, what was called previously Parvula, now it's Minotiflora, or here in Cluster Stigma. This is glistening. It looks like nectar. Some naturalists even said that there are glistening droplets of nectar, but it's not nectar. It's shiny, translucent cells at the base of the petals which mimic nectar. So to distract the pollinator from the real pollen, to keep it busy with trying to get this nectar while it's getting dusted with pollen. So if I have still some time. Another fascinating thing, so several Drosera species do not have nectar, but other plants have. And for example, this is a species of orchid. It's also nectarless, has a sweet scent. It's Telemitra antenifera, and it grows in huge numbers on these granite rock outcrops in Western Australia. But if you have a closer look, not all of these flowers are this Telemitra orchid. Here too, growing side by side. This is the Telemitra orchid. This is Drosera sapertella. Same flower size, same scent, so same floral bouquet, same orientation of the flowers. So this is what I mean with pollinator guilds. South Africa, Drosera cistiflora, and this is a species of Ornithogalum. I did not put them here for a photograph. They were growing side by side like this. Different location of members of the Drosera cistiflora complex is also in terms of flower shape, very diverse. Here, species of Psilocyne, and here another member of the cistiflora gang. We go further north, the cistiflora flowers get red, and here a Nemesia accompanying flower, very similar flower shape, color pattern. To understand this a bit more, I have to bore you with a family tree. You don't have to read all the names, just look at this. Some triangles are larger, the triangle size shows how much species we have, and these things show the relatedness. And I briefly have to introduce you to a thing called breeding system, which you all are familiar with. You probably know that some Drosera species, weedy things like Capensis or Spatulata, you can self with its own pollen. But they're also, these are so-called self-compatible species. They're also self-incompatible species, SI species. They're also SI species, which cannot be fertilized with their own pollen. They are self-sterile. But you need two different clones to get pollen, to get seed. So a pollen plant and the mother plant. And if you look at the distribution of these two patterns on the phylogeny, I marked in green all those species which can be selfed, and in red those which are self-incompatible, SI species. And it's very interesting. True SI species, I will come to these things later, but true SI species you exclusively find in Australia. This is a group of affinity, it is Australian clade, subgenus Ergolium, which is exclusively confined to Australia. Some Australian species can be self, but this is again a reversal to selfing. While as all the other species which are self-compatible, if we look at the flower diversity, these are the New World and the European diploid species. Almost all of them white, small flowers, some of them pink. The Brazilian tetraploids, section Brasilea, all of them similar flowers. Or here the African tetraploid species, very similar flowers. If you would look at the flowers only, you would say this is a single species. If you look at the leaves, you're of course convinced this is Drosera cuneifolia, Capensis, Eregrinii, Glabriscaba, no, not Glabriscaba, Glabripis, sorry, yeah, thanks. So they're very different in terms of their leaves and their overall habit, but the flowers are incredibly similar. Whereas the self-incompatible species, the SI species, they are very diverse, often many species sympatric, because they have they are obligate outcrossers, so they cannot rely on, well, if I do not attract the pollinator, then I just self and wait for the next year, maybe that's better, I'm better than attracting pollinators. They have to ensure it, and that's why we have such a floral diversity. This is a boost of driving speciation is that they cannot self. And also very interesting, we see this in pollen shape. From here on, all these selfing species, they have a pollen which looks like this. Very similar. This is the reason why you can make a hybrid between, let's say, Drosera graminifolia and Drosera montana or so on, or even an African Drosera spatulata and whatever. So, and even between the African, the Brazilian, or the European species, there's a famous hybrid created by a German breeder between Drosera venusta and Drosera anglica. So you can cross them all because they all have very similar pollen. So while the pollen of the self-incompatible species, it looks very, very different. And there we have plants which have a very similar flower shape. The styles are totally different. Look, this has little knobs here, little drumsticks. This has little, just like a piece of straw. And the pollen looks fundamentally different. And indeed, in Western Australian Drosera species, I can tell you the species of pygmy Drosera by a single style or a single pollen grain. And this is highly specific, and I assume it works like a key locker in a keyhole, the pollen on the respective style. I don't know of anyone who was ever able to cross different orange-flowered pygmy Drosera species. Now, for this group here, this is the winter-growing, summer-dormant African Drosera species. And again, some people say, well, I cannot get seed from my Drosera cistiflora or passiflora. It's self-sterile. It's not self-sterile, but it has the different floral organs ripe at different times. When the flowers of a member of the cistiflora gang opens freshly, then the anthers are open to present the pollen, but if you look at the styles, they are closed. They are sometimes even glued together. The same flower 3 days later, normally the pollen has passed away, moved away, and you see this is opened. It is closed here, and it's now opened like a claw or something, and now you can put the pollen on. So you have to harvest the pollen a few days before, and just if they open on the second or third day, the flowers are ripened. And this also goes along with a little color change. In some species it's more pronounced. You see it's greenish white in there, and as soon as the pollen is dispelled and they are in the female phase, they sometimes darken a little bit. Some water lilies do this as well. The pollinators, as I said, are monkey beetles, which feed on the flowers, get dusted in pollen. And the prey usually are very small insects, mosquito size. So there's no pollinator-prey conflict. In South Africa, I was catching some of these and placed them on the leaves, and they can remove themselves. They can escape from these traps within seconds. Just clean themselves, groom a bit, and then they leave away. So no pollinator-prey conflict. And we have these bicolored beetle-pollinated flowers in most winter-dormant South African species. Very interesting, also very species-rich group. So about prey-pollinator conflict, these are my last few slides. Some Drosera, they capture very large insects, like this Drosera intermedia, some damselflies in dragonfly size. And these are very few occasions. This is, for example, intermedia is pollinated by hoverflies, and there are some rare occasions where they trap hoverflies, but I didn't find a single pollen grain of Drosera on these hoverflies which are trapped. And this is my last story, a bit a curious story. In South Africa, in the Cederberg Mountains, you find this. This is a member of the Drosera trinervia affinity, and you find one large member of the Drosera cistiflora affinity. A lot of size difference, so flat rosetted Drosera trinervia, and this is, let's say, call it Drosera cistiflora for now. Larger flowers, upright stem with these leaves. These flowers are pollinated mainly by bee flies, which try to feed on this nectar. Again, this has some glistening areas here, which mimic nectar potentially, and they are not attracted by these sticky leaves here, but they go for nectar. And I found the same species of bee fly frequently trapped by this congener, by the sympatric growing speciation. So this is something for further study. It could be that the pollinator of one species is aiding as the prey. Here you see two of them trapped by the cystiflora as prey of a different species. So there could be competition for the same insect. One is feeding the pollinator of the other. So there could be competition in between different species. And with this, I like to thank you all for your attention. And if we still have time, I'm open for a few questions. And thanks to all the people who contributed photographs. Do we have time for a few questions? Yeah. 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 For a robot take. Yeah, there's no chance for a robot taking the. You probably have read that in Japan they try to. You mean a drone taking the? No, I don't. I need more motion activities. So my serious practical question is how cheap would such a thing have to be? You mean a drone thing, a robot which is pollinating plants?
Like a field camera that snaps and takes pictures of the deer when you're not there.
Yeah, now I know. Oh, okay, yeah. It does, unfortunately, it does not work. I tried with camera traps. And the problem is that, especially with Uticularia I tried. First of all, most of them, you either can have some with a sensor, with a laser sensor, and usually the pollinators are too small to activating the sensor. And if you make a very sensitive sensor to trap these small insects, you usually get photographs of flowers without an insect when there's some wind. So you take tons of photographs, any air movement when the flower is slightly moving takes it. The problem is that if you tried with webcam and everything, the pollinators are too small to be trapped on this. You just see a little piece of brown item moving there, but you can't identify the species. You have to use a macro photography, and I didn't find anything with a camera trap which works on these small scales so far. Okay, did this answer your question? Yeah. Okay, perfect. Yeah.
If I understood correctly on the Virigula, you said that locally the populations are genetically very uniform. Is that true across the board at different geographical locations? Are the Virigilas genetically different and that's just isolated uniformity, or it's all of them?
This depends. In Virigula gorgonias, there are actually only two major populations which are very similar. Dentata is very widespread, so we have genetically niched different populations, but Virigula is very old. It's a so-called paleoendemic, which has been sitting there for a long time. Probably lots of species got extinct. It probably went through lots of extinction. So what we probably have there is remnants of old populations. We also don't know if there might have been a pollinator which was able to pollinate Virigula, but which got extinct through the change of time, a few million years or so. Virigula is very old and very long sitting in this niche. So this is a very bad example also. It's very good, well studied. It's a very unusual thing. Yeah.
One more question, then we got to move on. Sorry, guys.
Yeah.
Why are some incompatible crossroads one variety of flowers find
Excellent question. Huh? So I repeat. He asked why the species which are self-incompatible, which are very diverse, restricted to Australia. Very excellent question. I have not a good answer. The thing is, the genus Australia the genus Drosophyllum comes from the southern hemisphere, and we have the early branching lineages, so the ancestral lineages we have in Australia, with the exception of Drosophyllum in South Africa. So, and it might be one of these ancestors which evolved this for whatever reason and didn't. But there are reversals. But I don't have an explanation. You could explain it because we have so much diversity on the same ground, so which always thrives, boosts speciation. To avoid probably, to increase genetic exchange, to be better adaptive to changing habitats or to get into niches. But it's an excellent question where I don't have a really good answer.
And branching off of that, I saw that the other very small branch of self-incompatible species in South Africa were quite far away.
Yes, and it's because in the Australian species, it's a pollen stigma incompatibility. It's a chemical thing or mechanical thing, and in the self-incompatibility in South Africa is totally different. They can self, but the organs are separated by time by dichogamy. So evolved 2 times independently. You can't compare these two.